Dear students, today we are going to discuss about the wonder topic structure of chromosomes. Chromosomes are the highly coiled and condensed form of chromatin fibers. During cell division, chromatin fibers condense to form chromosomes. Chromatin fibers are made up of nucleosome subunits. Nucleosome subunits comprises 40% of DNA molecule and 60% of histone octamers. The DNA molecule present in the nucleosome is very large and it is composed of two complementary strands. These two complementary strands wound around each other in the form of double helix. Each DNA strand is made up of nucleotide subunits which in turn consist of three parts: phosphate group, pentose sugar and a nitrogenous base. There are four types of nitrogenous bases found in DNA. They are adenine, guanine, thymine and cytosine. Adenine always pairs with thymine with two hydrogen bonds. Guanine always pairs with cytosine with three hydrogen bonds. Histone octamer. Histones are proteins that help in the coiling and packaging of DNA into structural units called nucleosomes. A single chromosome may have about 1,000,000 nucleosomes. I wonder how 2 meters of DNA is stretched inside 6 micrometer cell. If you come to the structure of chromosome, it consists of two sister chromatids. They are attached at the point of centromere. And each chromosome centromere is located at a particular site. It helps to attach spindle fibers during cell division. These spindle fibers helps to detach the chromatids during cell division. Genes. They are the specific sequences of nucleotides on a chromosome that encode particular proteins which express in the form of some particular feature of the body. Our height is determined by the genes and our skin color is also determined by the genes. Genes are the things that will transfer the genetic information from parents to the offspring. That's why we resemble our parents and grandparents. What is the need for new cells? So there are so many reasons for that. For growth, for repair, for replacement and for reproduction. Cell cycle. Cells are constantly going through the cell cycle. Once the cell divides, its daughter cells also just repeat the same. Cell cycle is nothing but it is the ordered sequence of events that occur in a cell to prepare itself for cell division. Cell cycle has two phases. One is interphase and the other one is M phase or mitosis phase. Interphase is the non-dividing phase of the cell cycle and the mitosis is the dividing phase of the cell cycle. Interphase. During this period, the cell grows and prepares for mitosis. It again includes three phases: first growth phase, synthesis phase and the second growth phase. In the first growth phase, RNA and protein synthesis will take place. The cell synthesizes various enzymes and nutrients that are needed later on for DNA replication and cell division. And the volume of cytoplasm slowly start to increase. Mitochondria and chloroplasts start to divide because they have their own DNAs. In the late G1, the cells either withdraw from the cell cycle and enter the resting phase or they will start preparing for the next cell division. In the synthesis phase, more and more DNA is formed by the process called DNA replication. The chromosomes are duplicated in this phase. In the second growth phase, RNA and proteins needed for cell division continues to divide and it is the shortest growth period. After the dividing phase gets completed, the newly formed daughter cells enter into the interphase again and the cell cycle goes on. Mitosis is the division of somatic cells, in which two identical daughter cells are produced by the division of one parent cell. It is called by the name equational division. Why? Because during the process, daughter cells receive an equal number of chromosomes. It is also called by the name somatic cell division because this division occurs in the body cells or somatic cells. It is helpful for growth and development. M phase consists of the following phases: karyokinesis and cytokinesis. Karyo means nucleus, cyto means cytoplasm, and kinesis means division. So karyokinesis is the division of nucleus and the cytokinesis is the division of cytoplasm during cell division. Karyokinesis occurs in four phases: prophase, metaphase, anaphase and telophase. The order of phases you can remember by remembering one sentence: please sit on the mat. Please, P stands for prophase, mat, M for metaphase, A for anaphase and T for telophase. During prophase, the chromatin fibers, contents and thick chromosomes are visible. The nucleolus and nuclear membrane disappear. A pair of centrioles duplicates. Spindle apparatus starts to form and the duplicated chromosomes start moving towards the equator of the cell. In metaphase, chromosomes are arranged on equatorial plane. Each chromosome gets attached to a spindle fiber by its centromere. In anaphase, the centromere divides and the sister chromatids separate from each other. Spindle fibers contract and pull chromatids towards opposite poles. In telophase, spindle apparatus disappears. The chromosomes become thin and turn into chromatin fibers. Nuclear membranes and nucleoli start to reappear. Cytokinesis. The furrow developed during the karyokinesis continues to deepen in the cell and it finally divides the cytoplasm, forming two daughter cells. In plant cells, instead of furrow, a cell plate appears at the equatorial plane. There is a slight difference between plant and animal mitosis. In animal cells, centrosomes are present, so asters are formed during animal mitosis. In plant cells, the centrosomes are absent, so astens won't be formed during plant mitosis. In animal mitosis, cytokinesis is formed by furrow formation, and in plant mitosis, it is formed by cell plate formation. Animal mitosis will occur in most tissues, but the plant mitosis occurs mainly at growing tips and sites. And both animal and plant mitosis are done for growth, repair and replacement. Meiosis. Meiosis is the cell division in which each daughter cell receives half set of chromosomes. Hence in meiosis, haploid daughter cells are formed from a diploid parent cell. Meiosis is also called reductional division because from diploid cells, haploid cells are formed. That's why it is called by the name reduction division. It consists of two successive divisions, meiosis 1 and meiosis 2. Let us discuss about the importance of meiosis. During meiosis, homologous chromosomes pair along their lengths. The chromosomes cross over at certain points called chiasma. At each chiasma, the chromosomes break and rejoin, trading some of their genes. This recombination results in genetic variation. So this is how siblings are different from each other in certain aspects. Finally, let us discuss the difference between mitosis and meiosis. Mitosis, it occurs only in somatic cells. Meiosis occurs in reproductive cells. Mitosis is only for growth and replacement, but meiosis is for gamete formation. Mitosis occurs continuously throughout life, but meiosis occurs only in reproductive age. Because of mitosis, 2 daughter cells will be produced, but by meiosis, 4 daughter cells will be produced. And full set of chromosome is passed on to each daughter cells during mitosis. But in meiosis, only half the number of chromosomes is passed on to each daughter cells. In mitosis, single nuclear division will take place, but in meiosis, 2 nuclear divisions will take place. This is the homework for everyone. Pretend your friend was absent from the class today. Write about what you would say if you had to explain the lesson to your friend. Thank you.